is a good time to have lecture and listen our national president, Professor Mande sir. So in this region and our VVM internship, which we have created last year for our national camp winner, जो अपने नेशनल कैंप विनर में जो हैं उनके लिए जो हम लोगों ने एक नया प्रोग्राम बनाया, जिसको कि हम लोगों ने इंटर्नशिप सीजन इंटर्नशिप के नाम से दिया, जिसमें बच्चों ने जुलाई में एक सप्ताह का इंटर्नशिप किया और बीच बीच में हम लोग लेक्चर करते रहे हैं इन बच्चों के लिए। उस पूरे एक साल की जो यात्रा है, उस यात्रा के जो समापन कह सकते हैं लेक्चर में, हम लोग आखिरी लेक्चर में आ रहे हैं अब इस पड़ाव के, और इस आखिरी पड़ाव के लेक्चर में आज हमारे बीच में प्रोफेसर शेखर मांडे जी हैं। वो आज इस लेक्चर के लिए हमें समय दिए हैं, जो हमारे विज्ञान भारती के नेशनल प्रेसिडेंट हैं। सो शेखर मांडे जी का मैं नमस्कार करता हूं। स्वागत है सर आपका आज के इस ऑनलाइन लेक्चर में। हमारे वीवीएम के नेशनल कन्वीनर हैं डॉक्टर प्रोफेसर प्रशांत कोदगिरे जी, आईआईटी इंदौर से प्राध्यापक हैं। स्वागत है आपका सर। इस प्रोग्राम में, आज के कार्यक्रम में हमारे जो इंटर्नशिप के कन्वीनर हैं सुधाकर जी, सुधाकर जी, डॉक्टर सुधाकर जी, सो मैं उनका स्वागत करता हूं। और हमारे इंटर्नशिप प्रोग्राम के मेंबर हैं उमेश क्षीर सागर जी, इनका इन प्रोग्राम में आज ये सब लोग हैं यहाँ पे आपके साथ। और पूरे साल भर जो हम लोगों का ये इंगेजमेंट प्रोग्राम रहा है बच्चों के साथ, बहुत सारे ऑनलाइन लेक्चर्स हुए हैं, कुछ एक्टिविटीज बच्चों को दी गई हैं, एक सप्ताह का रिगरस हम लोगों का ट्रेनिंग पीरियड रहा है आईआईटी में, दिल्ली में। उसके बाद आज जो है हम लोग एक लेक्चर के रूप में प्रोफेसर मांडे जी को सुनने के लिए हम लोग यहाँ पे हैं। और आगे हम लोगों का इस साल जो 2022-23 का जो नेशनल कैंप होने वाला है वो 20 और 21 मई को है। इसके बाद जो हमारे पास बच्चे आएंगे उनके साथ लेकर के हम अगले साल का फिर से ऐसे ही एक रिगरस प्रोग्राम चलाएंगे। और मुझे उम्मीद है, विश्वास भी है कि इस बार जितने बच्चे हमारे 90 जो नेशनल कैंप विनर हैं उसमें से मैक्सिमम बच्चों ने पार्टिसिपेट किया होगा और वो बच्चे हमारे नेशनल कैंप में हमारे पास आने वाले हैं। सो विद दिस वर्ड्स आई विल इनवाइट आवर नेशनल कन्वीनर प्रोफेसर प्रशांत कोदगिरे जी टू से फ्यू वर्ड्स एंड इंट्रोड्यूस आवर स्पीकर प्रोफेसर मांडे जी। प्रशांत जी।
बहुत बहुत धन्यवाद ब्रजेश जी। आज हम लोगों का सौभाग्य है कि देश के जाने माने और बहुत वरिष्ठतम वैज्ञानिक डॉक्टर शेखर मांडे सृजन इंटर्नशिप की श्रृंखला में आज टॉक देंगे। जैसा कि ब्रजेश जी ने बताया कि डॉक्टर मांडे वरिष्ठ वैज्ञानिक भी है, लेकिन हम लोगों के लिए एक इमोशनल कनेक्ट ऐसा है कि मांडे सर विज्ञान भारती के माननीय अध्यक्ष भी है, प्रेसिडेंट भी है। इस कार्यक्रम की फॉर्मेलिटी के लिए मुझे परिचय देने के लिए बुला है। बहुत संक्षेप में मांडे सर का परिचय देना चाहूँगा। Mande sir completed his MSc in physics from University of Nagpur, and then he moved to one of the very prestigious institutes for his PhD, Indian Institute of Sciences, Bangalore, and he got his PhD in molecular biophysics. Subsequently, he moved to Europe, Netherlands, for his postdoctoral studies, and then he returned and joined one of the very prestigious, eminent institutes, one of the CSR labs in Chandigarh, Institute of Microbial Technology. He worked there and then subsequently moved to yet another institute called as Center for DNA Fingerprinting and Diagnosis. Short form of that is CDFD. That is in Bhagyanagar, Hyderabad. After that, he was also in an administrative role. He was director of another very prominent institute in Pune, National Center for Cell Sciences. After his directorship at NCCS, he went to Delhi as Director General of CSIR. And then currently, Dr. Mande is Distinguished Professor in Savitribai Phule University. Dr. Mande works on different aspects of structural biology with respect to Mycobacterium tuberculosis. So he is a famous structural and computational biologist. He has got many awards. Just to name a few, he got B.M. Birla Young Scientist Award. He got highly reputed Wellcome Trust International Senior Fellow Award, and so-called the best scientific achievement in India is getting Shanti Swarup Bhatnagar Award. Mande sir got that award in 2005. Subsequently, he became fellow of different academies in India. He got BC Guha Memorial Lecture Award, BK Bachawat Memorial Lecture. He also got Bharat Asmita Award, and then HK Firodia Vigyan Bhushan Award. So uh, we are privileged to have a really great scientist today who is going to talk about very fundamental work. Title of his talk is Molecules of Life, and he has precisely chosen this because of the audience which we have. We know that you have graduated from class 6 and you are now from class 7 onwards to class 12. So I hope you're going to enjoy this interaction with a great scientist of India. Uh, over to Brijesh ji. Thank you very much for giving me opportunity to introduce today's speaker.
थैंक यू प्रशांत जी। अभी हमारा समय हो रहा है कि हम सर को बिना समय गंवाए सर को सुने। सर के सुनने के बाद जो भी हमारे ऑडियंस हैं, जो हमारे छात्र हैं, सुन रहे हैं, 6 to 8 क्लास के रहे हैं, इन सबों के मन में जैसा भी कोई प्रश्न हो, कोई क्वेश्चंस हो, आप यूट्यूब के चैट बॉक्स में वो क्वेश्चन हमको लिख के अपने नाम के साथ, क्लास के साथ आप दें। हम इस सर के लेक्चर के बाद एक एक प्रश्न हम आपके तरफ से पूछेंगे और उसका जवाब आपके पास तक आएगा। तो आप ये ध्यान रखें, जो भी आपके मन में प्रश्न आता है, जिस तरह से आप लिख सकते हैं उसको चैट बॉक्स में लिख करके हमको भेजें। हम उन प्रश्नों के जवाब सर से आपको दिलवाएंगे। तो इसी के साथ मैं मांडे सर से आग्रह करूंगा, सर प्लीज स्टार्ट द सेशन।
बहुत धन्यवाद ब्रिजेश जी, धन्यवाद प्रशांत जी, और सभी बच्चों को मेरा हार्दिक नमस्कार। हम लोगों को बहुत गर्व है आप सब युवा बच्चों को पर, जिन्होंने हमारे विज्ञान की विद्यान मंथन में एक सहभाग लिया। और आज आप जिन जिन विद्यार्थियों को यहाँ पे पुरस्कार प्राप्त हुए हैं, मैं सभी का हार्दिक अभिनंदन यहाँ से करना चाहता हूँ। आने वाले आधे घंटे में मैं कोशिश करूँगा कि लिविंग सिस्टम्स किस तरह बनी होती है, उनको हम किस तरह समझ पाते हैं वैज्ञानिक दृष्टिकोण से। तो कोशिश करूँगा आपको समझाने की। अगर कुछ समझ भी ना आए इधर उधर की चीज, तो जरूर प्रश्न पूछिएगा। प्रश्न देने के लिए तो मैं बहुत खुश होऊंगा आपके सामने में। तो मैं शुरू करता हूँ कि अपना टॉक का टाइटल है मॉलिक्यूल्स ऑफ लाइफ, और देखता हूँ कि अगर मैं स्क्रीन अगर शेयर कर पाया हूँ तो आई होप ऑल ऑफ यू आर एबल टू सी द स्क्रीन। प्रशांत जी, आप स्क्रीन देख पा रहे हैं मेरी?
यस सर।
चलो, तो मैं आपको बताना चाहूंगा कि लिविंग सिस्टम्स हैं कि व्हाट काइंड ऑफ मॉलिक्यूल्स मेक अप लिविंग सिस्टम्स, एंड दैट्स एसेंशियली व्हाट आई वुड लाइक टू टेल द हाई स्कूल स्टूडेंट्स कि मॉलिक्यूल्स किस तरह बनते हैं, उसके से लाइफ का उत्पादन किस तरह होता है। तो बेसिकली अगर इफ वी लुक अराउंड अस एवरीवेयर, we look at many things. We look at let us say stars, we look at the sky, we look at trees and plants around us, we look at animals, we look at insects, we look at mountains, we look at rivers, and all of them have some kind of a size. And naturally many people actually start asking question ki what kind of sizes these things have, and if they move, what kind of motion or at what level they move. For example, when we walk at a normal speed, we take a few steps per second, right? Maybe in a minute we take 2 or 3 steps, or maybe 5 steps. But at the same time, if bacteria move in a solution, how fast do they move? And that is actually what I have shown you here in this one. I also tried to show you what are the sizes of different entities in nature. For example, water is very tiny, but our cells by which we are made up of are somewhat larger. And this is exactly what we are going to see, that how actually these things are and how do we understand some of these. And when people began looking things around us, many people actually were very curious. And Indian history, of course, is very rich for centuries. How curiosity led to development of new concepts in what we understand as in science today. But one of those concepts was, if you have two different colors of flowers, and it was Mendel who observed this through his kitchen window, that there are two plants of peas, chickpeas, and one of them had a flower colored purple and another had a white color flower. And if he crossed these two plants, he got only the purple color flowers, but the progenies of this had one white color and three this thing. So that is a natural curiosity that Mendel had, but similar natural curiosity is there amongst many people. And as I said, India is very rich in people who have contributed enormously to science. So these kind of observations eventually lead to the question: how does this happen, and why does this thing happen? Like in the first generation, you have only purple flowers, but in the second generation, you have one white flower and three purple flowers. And why so? And that is the question that actually we ask. But let us go back even more fundamentally and ask a question: uh, what are actually living systems and what are not living systems? And this actually question is much more fundamental than what I've told you Mendel's questions. For example, if you look at this slide, the grass, as we understand, is a living system. Stones are not living system, non-living system. If you look at any of the kitchen utensil, that's not a living system. But if you look at a cactus plant in a some sort of a desert, we know that it's a living system. A tortoise is a living system, but a dead wood, what is shown here, is not living. So we must understand what is the major fundamental difference between these two. So before we come to the question about the plant that I showed you in the previous slide, the purple color flowers and white color flowers, we must ask ourselves the question that what is living system and is what not living system. And there are many answers to these questions. If I ask you, you will all start answering many different things. But what we can say broadly are hallmarks of the living systems. One of that is they can reproduce themselves. They can make their own kind of entity, the children who actually are also living, right? So, ability to reproduce is one of the hallmarks of the living system. Similarly, living systems, when any child or any small plant grows, it actually starts very small and then grows very big and develops. Right? Develop, when I mention, is actually the life starts with maybe one cell, and that cell divide, and eventually we form hands and legs and nose and eyes and so on and so forth. So every living system, whether it be it animals or plants or something, they grow and they develop. Another hallmark of the living system is the ability to obtain and use energy. Right? I mean, when we eat food, essentially the food eating is for getting energy. When plants trap solar energy, that is essentially to conduct photosynthesis in the plants. So the living systems have the ability to obtain and use energy. And if you look at the same thing about the stones or whatever, they don't have this ability. Same thing, stones don't grow and develop. So one of the major differences between living and non-living systems is this particular one. And more importantly, living systems can respond to environmental fluctuations. So all these, and there are many other answers that can be given to say these are the things which characterize living systems versus non-living systems. And let us actually look at how this arises at a very fundamental level. The second thing we observe is that when the living systems reproduce, remember in the previous slide I showed you that one of the hallmarks of living systems, they reproduce themselves. And when they reproduce, they create offsprings of their own. For example, a tiger, when it reproduces, it makes a baby tiger. A monkey, when it reproduces, it makes a baby monkey. Right? the question that we asked in the first slide about the pink flowers and white flower, we saw that that question eventually leads us to the fact that tigers' babies are only baby tigers and monkeys' babies are monkey babies. It never happens that a monkey baby is a tiger baby and tiger baby is a monkey baby. It never happens that way. And when we say reproduce ourselves, we essentially make copies of ourselves, something like this. To give a more example that connects with all the students, younger students here, and we also say that the children usually also resemble their parents. And example, if you see on the slide here, that son of Mr. Amitabh Bachchan looks almost the same as Amitabh Bachchan. Right? I mean, these are the kind of things that living system also have. That not only we make our own babies, which resemble us, but also look very much like us. So these are the hallmarks of living systems, and we actually want to say that we want to understand how all this happens. I mean, one of the questions that I asked first, that how does all this happen at a very small level, at a molecular level. And one of the first things that people notice when they were trying to understand what are living systems and what are non-living systems, for example, how do you distinguish a stone from a plant? People realize that plants and animals, they are made up of cells. When they looked under microscope, they look at cells, and some of you may have done this experiment, looked at onion or whatever under a microscope, and you actually look at a cell, beautiful cell. But people actually also realize that there are very tiny objects which are called as bacteria. They're very, very tiny. And they and our own cells, like plant cells or animal cells, have some major differences among themselves. So people actually started calling the tiny things like bacteria and all as prokaryotes. You don't have to remember the name right now. Just remember that two kind of the organisms that we'll talk about. Prokaryotes are the bacteria, and eukaryotes are people like us or animals or plants. They're eukaryotes. And this was actually discovered under a microscope that in a eukaryote, the cell, there are compartments and there are many different what we call as these things as different organelles. For example, this is mitochondria, this is nucleus, and within the nucleus you have the DNA. And let us actually look at the molecular level once again how this actually happens. So there are different kinds of cells, different kind of organisms, very small tiny ones that can be seen only under a microscope, things like bacteria, or there are larger organisms which have multiple cells in them, so multicellular organisms or multiple cells in them, such as ourselves, humans, animals, plants, or something. And let us actually start looking at the molecular level how they look. And when people actually started trying to understand how these cells are made up of, which kind of atoms, the first thing that people notice, and it is just amazing, that all life forms are made up of very similar atoms. If you take a bacteria, if you take a virus, if you take a small parasite, or if you take a plant, you take a dog, or you take a human being, or whatever, all the cells at the fundamental level are made up of very similar kind of atoms. For example, carbon, hydrogen, nitrogen, oxygen, sulfur, and phosphorus make up about 99% of cellular weight. But beyond atoms, when we go to molecules, all our cells are about 70% water. And that's why water is very, very important molecule of life. But we also have inorganic ions, such as we have phosphates, we have sulfates, we have calcium, we have iron, and so on and so forth. They also exist in living forms. There are also sugars, amino acids, and all. And let us actually look at some of these, how they make up life. So remember, we are trying to distinguish living systems from the non-living systems. And what characterizes the living systems? And what we saw already, that living systems have cells. We could see the cells under microscope. And at the molecular and atomic levels, living systems are made up of molecules such as this. And let us try to actually look at some of the molecules more closely. For example, as I said, water is one of the most important molecules of life. Without water, there will be no life whatsoever left. So all the modern life forms that we see around us, and once again let me say, from bacteria to parasites to plants or animals or human beings, all the living forms, whether it be it fish or whether it be it any kinds of exotic plant, all of them have important ingredients, and water is by far one of the most important ingredients. And water is made up of two hydrogen atoms and one oxygen atom, as all of you know. And water has many important properties. And one of the important properties of water is that it has a high heat of vaporization. Okay? What it means is that the term high heat of vaporization means that the amount of heat needed to turn a given amount of water into another form, in this case from liquid water to vapor water, is enormous. It's very high. So the heat of vaporization, water can absorb lot of heat before it converts itself from liquid to gas or vapor, water vapor. And that's a very, very important property in all the living systems. Why? Remember, I told you in one of the first slides that living systems also respond to environmental fluctuations. Remember this? I told you we can respond to environmental fluctuations. For example, right now is month of May and it is getting very hot in different parts of the country. But come month of November and December, it will be very cold. And that actually has a change in environmental fluctuation. The temperatures at that time are very different. But we can sustain these changes in temperature, we can sustain. And that's precisely because water, our body uses water as a temperature buffer. As I said, all our cells have 70% water, and water is a major temperature buffer. Let me give you an example. When all of us, if we do some rigorous exercise, let us say we go out and run for a long time, or we play a game for a long time, such as tennis here. What I've shown you is the tennis player Adre Agassi, who's playing tennis. When we do this thing for a while, short while, we realize that we start sweating. And why do we sweat? We sweat because we have done so much exercise that our body is getting heated up. And when the body gets heated up, it is important to cool the body down. And to cool the body down, we actually start sweating. And the sweat comes on the surface of our skin and then vaporizes. And when it vaporizes, it releases very high heat. See, I told you heat of vaporization. So amount of sweat that actually eventually vaporizes helps the body to cool down. So whatever heat the body might have generated is now cooled down by the main of sweating, and the sweat actually eventually evaporating, and that cools the body down. And that's precisely what I said. Water acts as a temperature buffer against environmental changes. And there is only one property of water that I told you. There are many properties of water that we can discuss which are important for the living systems. Now, these properties the non living systems do not possess. And therefore, water we can consider as one of the most important molecules. But let us look at some other molecules of life. And people actually started looking at other molecules of life and very soon realized that all different living systems, bacteria, plants, animals, etc., they are made up at the fundamental level by the same kind of molecules. And it is amazing. How can all living systems be made up of same kinds of molecules? And I'm going to give you examples of those kinds of molecules. But believe me, all living systems on the planet are made up fundamentally of very similar kind of molecules. That suggests that several years ago, a few billion years ago, there must have been only one life form that came into being, and that life form, when it grew and when it reproduced, it gave variety of life forms what we see today. And to prove this particular fact, two gentlemen, one called Miller and one called Urey, they actually in 1953 did an experiment with the starting material of methane, ammonia, hydrogen, and water, and put this particular mixture in a flask and then allowed sparks to go through the flowers. Remember, in the early atmosphere of the earth, there were a lot of sparks that were flying. And when they did that and they condensed whatever was being formed in this particular chamber, when they condensed, they found it had several compounds which had formed. And all these compounds are reminiscent or they resemble what we see today as the molecules of life. That is phenomenally important. That is so cool that with this experiment they could see that molecules have arisen in the very beginning of our lives in the sense the living systems on the planet, the earth, and they continue to be the same kind of molecules that have survived, and they make up the entire living system spectrum today. And let us have a few examples and then I will stop. For example, the genetic content in all our cells, once again, in all living forms, bacteria, viruses, plants, animals, is something called the DNA, right? Or at times it's RNA, but also DNA. And the DNA is essentially made up of one sugar molecule, one base, and one phosphate. Our bodies, all day to day jobs are conducted by what we call as proteins. They are made up of amino acids. Also, our body requires sugar for its own metabolism. We use sugar, break it down, and generate energy out of it. There are different kinds of sugar that exist. And all the cells are enveloped in what we call as a cell membrane, and that also actually are forms of molecules that exist. All these kinds of molecules are common to different living systems or living forms. And let us look at all the four little briefly, how they are actually and what kind of characteristics they have. For example, cell membrane, if you take, I showed you a cell some time ago on my slide. The cell essentially forms the boundary of what is there inside the cell or what is there outside the cell. It's kind of a cocoon, a very small cocoon. Inside the cell, you have, as I showed you, in a eukaryotic cell, there are mitochondria, there is nucleus and all. But even within the bacterial cell, there are nucleic acids, there are proteins, and so on and so forth. But it's important to keep these things within the cell and not let them go out. And that job is done by the lipids, what we call as lipids. A typical lipid looks like this. It has several carbon atoms. What are shown here in black are these carbon atoms. Okay, and it is CH2, 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 CH2 like this, both the sides. Now, those who understand chemistry, or those of us who actually can look at chemically, it turns out that this long chain of CH2, 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 CH2 makes it very hydrophobic. That is, it does not like water. And I remember I told you, water is a very, very important molecule of life. So if the cells have, they're surrounded by this lipid molecule, obviously what is water inside the cell cannot escape very easily. In fact, water cannot escape. It has to be only transported by very specific mechanisms, if at all, to outside the cell. But outside the cell, on the beyond the boundary of the cell, we also have what we call as the phosphates, right? What you see is a PO4. We have a glycerol, and we have at times choline or ethanol or serine or something like that at the cap of it. Now this is hydrophilic, this part. That is outside the cell. This likes water. In the membrane, it does not like water. So essentially, we have formed a boundary on the surface of the cell by molecules such as this, such as a membrane lipid, which, when projected towards the cell, has a very hydrophobic, that is, water hating. The hydrophobicity means that does not like water. And outside the cell, you have that likes water, is a polar thing. So therefore, you have two kinds of boundaries that are formed, one in which water cannot enter, other in which water can interact very freely on the outer side. So outer side of the cell, water can interact very freely. And the membrane then would something look like this. So you see here, these long carbon chains are like this, and there are two layers which are formed. The polar layer is outside the cell, and this polar layer is inside the cell. So you have what we call as a bi-layer membrane. The two layers which are formed by the same molecule. So this lipid, it formed two layers facing on opposite sides. On the outside of the cell is a polar part. In the inside of the cell is a polar part, but in between it's a very thick hydrophobic, or the part which does not like water. And I told you that water molecules cannot go through the membrane unless they are assisted by some water channels across this thing. So there are transmembrane proteins that can carry out many jobs, such as carrying water from inside to outside, and so on and so forth. So the lipid layers are formed like this, and they protect cells from everything that is there inside the cell, from what harmful thing may be outside the cell. So the job of the membranes is to form a boundary around the cell. Now, how do we get actually energy, and how do we transfer information? And I told you that our energy we get from sugars, and typically what we have are sugars such as glucose, such as fructose, and such as sucrose, which is actually a disaccharide of glucose and fructose. They are all very important because our body can take up the sugar and eventually convert the sugar, break it down, and convert it into a molecule called ATP. And ATP is the molecule which stores energy in our cells. So sugars are very important to derive energy from. But the different kinds of sugars, they're also important to form the backbone of the DNA. Remember I told you a molecule called DNA or the RNA, the deoxyribonucleic acid or the ribonucleic acid. Both of them are the molecules of information. And it's beautiful. How do the molecule of information form? You have a sugar which has 5 carbon atoms, 1, 2, 3, 4, 5, or rather 1, 2, 3, 4, 5. And on one of the carbon atoms, we have a base. On another carbon atom, we have a phosphate. On one carbon atom, we have a hydroxyl. The bases in nature can be of multiple types. Typically, in a DNA molecule, you have adenine and guanine, and you have cytosine and thymine. These 4 bases typically occur in RNA, in DNA, but in RNA you have uracil. Okay, so essentially, what you have is instead of thymine, you have a uracil in RNA. So these are the 4 essentially nucleotides that form our DNA. And what our DNA does? DNA, the bases and the sugars and the this thing are all joined back to back to this. So this is one kind of a building block of DNA molecule, and on this phosphate you will have a covalent bond with this hydroxyl, and therefore it can form an infinite chain like this. There's a phosphate here, there's a hydroxyl of another sugar atom, and that sugar atom then attaches here, and another phosphate and the hydroxyl bond that forms there, and so on and so forth. Many different bases together can form a DNA molecule. So for example, you have one strand of DNA like this, going like this, has many bases. The bases what you see are inside here. Typically in human DNA, there are 3.3 into 10 to the power 9 bases. So if you write 9 zeros in front of 3.3 is what you will get, those many number of bases what you see here. And the bases are such that only one kind of a base can pair with another. You see here there are 2 strands of DNA. There is one strand which is going like this. There is one strand, and there is another one which is going like this. And in fact, these two are going in opposite directions. For example, if this strand is going from bottom to top, this strand is going from top to bottom. But what you see here in between is a pair. The 2 kinds of bases. You see a yellow and a red and yellow, and so on and so forth. They all form pairs. And only 2 kinds of pairs can be formed. If you have one side adenine, base something like this, it can only form a pair with thymine. Instead, if you have a guanine, it can form pair with cytosine. So the A and T can form a pair, or G and C can form a pair. So in the DNA molecule, if you have adenine, you necessarily have a thymine here. And if you have a G here, you necessarily have a C here. So what we can see now beautifully, that bases will start coming in order, maybe A, T, G, C, C, G, G, A, T, or something like that. But on the opposite strand, exactly opposite base would be there. So if there is a A here, there will be T here. If there is T here, there is an A here. If there is a G here, there is a C here. If there is a C here, there is a G here. So the sequence of the bases is very, 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 very crucial. The sequence in the base with which the bases occur is essentially the information what we have. The information that we pass on from generation to generation, such as I showed you, a tiger can form a tiger baby or a monkey can form a monkey baby. Essentially, what they were doing is that they were copying this information. There is the sequence of the bases that were coming on the DNA is being copied into the offspring. And how does it get copied? There's a double-stranded DNA. The two strands separate out, right? This DNA, the two strands will separate out. There's one strand, there's another strand, and very soon in the cell, it will start synthesizing a strand which is complementary to this, and another strand which is complementary to this. And eventually, when we are copied, both the complementary strands, the green ones, the DNA actually would have made two copies of itself. Starting from one DNA, it would have made two copies of itself, which are identical exactly to the parent DNA. So you see how information from one DNA molecule has been passed on to two DNA molecules. And in turn, these two DNA molecules once again will split like this and form four DNA molecules. And the four DNA molecules then will form 8 DNA molecules. And 8 DNA molecules will form 16 DNA molecules. But each time, the sequence of bases which are coming on the strands is identical. And that's exactly what all the living systems do. When they make a copy of themselves, they make a copy of the DNA exactly as it occurs in the parent, also occurs in the offspring. And that's how information is transferred from generation to generation. And that's why a tiger's baby is a tiger and a monkey's baby is a monkey. And that's why we all resemble our parents, because we have inherited characteristics from our parents. And that key is in the DNA molecule. But there are other molecules of life which are very important and which help our cells continuously in some functions. For example, I told you a water molecule has to be taken from inside of the cell to outside of the cell. There's a water channel for that. And the water channel sits in the membrane. And the water channel is a class of molecules what we call as proteins. And there are many, many different kinds of proteins in our body. There are proteins like hemoglobin, there are proteins like antibodies, there are proteins like insulin, there are proteins like water channels. All these proteins have very well defined tasks to do. And just one or two tasks actually I will define here. And proteins also are polymers, just like what we saw, the DNA is a polymer. Proteins also are polymers, and a polymers, the basic fundamental building block is what we call as an amino acid. Amino acid because it has an amino group, has an acid group. But then, unlike the DNA, which had only four kinds of bases, A, T, G, C, proteins have 20 different kinds of what we call as side chains. 20 different kinds. All right. And once again, the amino acids can condense together and form polymers just like the DNA molecule form, a single chain polymer. So this is one amino acid, there is another amino acid, there is yet another amino acid, and so on and so forth. So all of them actually can form a polymer. And a polymer once again has a sequence. Just like what we saw in the DNA, there's a sequence of ATGC. The proteins also have R1, R2, R3 or something like that. And these are all 20 different kinds of amino acids. So that sequence is there in proteins as well. And that sequence allows proteins to form a folded structure. And that folded structure is the one which is most important. And then that actually gives rise to many, many, many different functions. Like hemoglobin has a function of carrying oxygen from our lungs to our fingertips and so on and so forth. That's the function of hemoglobin. But in the fingertips and all, there is carbon dioxide which is accumulated, and it has to be brought back to the lungs. That's what hemoglobin does. But they are very, very different complex protein assemblies, and proteins can also form assemblies with DNA, with RNA, and so on and so forth. And what you see in the slide here is one of the most fascinating assemblies of living systems. It's called the ribosome. And it does such complex reaction. It's just unbelievably complex. The ribosome, the kind of things it does, and that makes life so beautiful. Ribosome essentially synthesizes proteins in our cells. Given a sequence of a gene, for example, a hemoglobin gene has to be converted into hemoglobin protein. Ribosomes help to do that. And just absolutely fascinating. It's just amazing how they do that. One of the most complex chemical reactions assemblies such as this do. But proteins also do many other functions, and those functions may not be as good. For example, here, what I show you here is the flu virus. What we call is influenza, that virus. On the surface of the virus, there are two kinds of spikes. One is called hemagglutinin, one is called neuraminidase. And it is a protein. Hemagglutinin is a protein on the surface of the virus. Neuraminidase is a protein on the surface of the virus. But when virus senses our cell surface, our human cell surface, it is in this particular form. But very quickly, when it actually binds to our cells, it converts into this form. That is yellow and blue, what you see, now forms a straight line here. And that essentially penetrates into our cell. So virus binds to the cell surface, human cell surface, and then this actually forms a long needle kind of a thing that allows virus to penetrate into our own cells and makes an entry. And once it enters our cells, it starts very rapidly making copies of itself. Remember the coronavirus, what we call as COVID-19? It is also called as the SARS-CoV-2. This virus can enter our lung cells and start making copies of itself very rapidly. And in literally no time, it makes millions and billions of copies of itself inside our own cell. And that essentially puts a lot of system, on our overall health. And that's exactly what viruses such as influenza and SARS-CoV-2 do. But the proteins that are important here are these proteins such as the hemagglutinin in influenza virus, or it's called a spike protein in the coronavirus. They help in binding to our own cell receptor and gain an entry into our own cells. So what I wanted to tell you was there are many different kinds of molecules of life. We saw four examples. We saw lipids, we saw carbohydrates, we saw nucleotides, and we saw proteins. All of them help in sustaining life. And in all living systems, whether it be bacteria, fungi, parasites, plants of different kinds, very tall plants, very short plants, algae, fungi, animals of all different kinds, whether it be it elephant or whether it be it an ant, insect like an ant, or whether it be it human beings, we all share the same molecules of life. An ant has an DNA, an elephant has an DNA, humans have a DNA. Ants have proteins, we have proteins, and plants have proteins. So all living systems share the same kind of molecules of life, and that makes life a beautiful experience. And that's all of us are going through the experience of our thing, what we call as the living system, as we call as the life forms, as what we are experiencing right now. And it's a beautiful experience. Let us enjoy it and let us sustain it for many, many generations to come. Thank you all very much.
अ, नमस्कार, धन्यवाद सर। बहुत ही रोचक, और मैं बायोलॉजी का नहीं हूँ, फिर भी मुझे वो सारी चीज़ें बहुत ही साफ-साफ समझ में आ रही थी कि क्या कैसे है, और मुझे उम्मीद है कि जो हमारे बच्चे अभी इस शो में लाइव देख रहे हैं, और अपना ये यूट्यूब का है जिसको कि बाद में बहुत सारे बच्चे देखते हैं उसको, और देख कर के समझते हैं, और बार-बार इसको देखा जा सकता है। ऑनलाइन का एक फ़ायदा ये है कि रिकॉर्डेड रहता है, और जब भी हम लोग यूट्यूब पे चाहें उसको देख सकते हैं। तो बहुत ही रोचक ये एक रहा। छोटे-छोटे 2-3 प्रश्न हैं अ, यहाँ पे बस वो आपसे मैं एक बार बच्चों की जानकारी के लिए पूछ लेना चाहता हूँ। तो एक पहला प्रश्न है सर, हाउ डू यू फाइंड एक्स्ट्रा टेरेस्ट्रियल लाइफ ऑन अदर प्लैनेट्स?
हाँ, तो ऐसी बात है कि आई सेड कि अर्थ पे जो लाइफ है,
जी।
वो जो उसके मॉलिक्यूल्स मैंने आपको बताया, डीएनए, प्रोटीन्स, लिपिड्स, शुगर्स वगैरह, it is not necessary that these are the only kind of molecules that form life. They form life on the earth, which is true, but you have to have self-replicating molecules in the first place. And it may be possible that on some other planet there may be other kinds of molecules which are self-replicating, and maybe those kind of self-replicating molecules are given rise to different living systems on that particular planet. But on today's date, we have no answer to that. We don't know what kind of molecules exist on other planets. So we have to assume right now that hopefully a similar kind of accident that happened on the Earth when we formed DNA and other molecules, the similar kind of accident would have happened also on other planets. But on today's date, we do not have answer for this question.
Okay, sir. One again small question. What are the analytical techniques to detect the presence of these molecules to detect life on other planet? Do you have any techniques we have?
Yes, so basically, what we do, you have to essentially look at what kind of molecules we can characterize very easily. The chemical structure of the molecules, whichever it is, can be characterized. One has to essentially show that those kind of molecules are self replicating. So self-replication is very key thing. So hypothetically, some extraterrestrial life exists, and we come in contact with that. The first thing that will have to be shown is that it has a property of self-replication. So we'll have to set up a controlled experiment to give conducive kind of an atmosphere to that system so that it can start replicating. And it replicates and it forms a copy of its own. Then we can say that indeed that's also a living form on that particular planet.
Okay, sir. Thank you. One, I think this is the last question. Molecules of all the material things, animal, planets, or whatever we can say, yeah, this is biological, that is geological or botanical. For all of them, these molecules are same. That is made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus, as you mentioned. How they know that they have to combine to form a animal, particular animal, a human being, or particular planet? That from where, like a human, you say that a lion produces baby lion. In this person, male female, they are producing this male female. How this molecule they know that I have to combine in this particular shape or order to give a baby lion or baby tiger or baby what is called dog or whatever that is? So that is the
So
question. Yeah.
So the mechanisms of reproduction are very well worked out. Essentially, what happens is that, I mean, in an animal, an egg from a female fertilizes with a sperm from a male, and then actually what happens is that through a series of steps, it starts forming copies of its own DNA. So the basic building blocks of DNA exist inside the cell, or cell can make them. What we called metabolism some time ago, cell can make actually those kind of molecules within themselves using some very complex enzymatic machinery. And those building blocks of DNA are transported into the nucleus, and the DNA can start forming copies of itself. Once it comes to a particular stage, that DNA has formed copies of itself, and these copies can be distributed to daughter cells. The cells divide, and one cell makes identical copy of itself, two identical copies of itself, and those two identical copies of cells then make four copies and then eight copies and so on and so forth. So at the cellular level, the cells keep dividing until a fully grown animal is formed, right? I mean, that's how different organisms would be formed. Even in a plant, the same thing does. The bacteria also does the same thing. That cells eventually divide, but for division of cell, the machinery that is required to make copies of the molecules, that machinery already exists, all the enzymes and building blocks and all. And when we say that we can actually living forms are characterized from non-living forms by acquiring nutrients and converting that into energy and grow and metabolize, is essentially all of these combined together, is we can actually start dividing ourselves and make progenies of ourselves.
Okay, so thank you for this simple answer of this difficult type of questions. So मैंने जो किया और फिजिक्स का विद्यार्थी होते हुए मुझे जितना अच्छे से बायोलॉजी समझ के आया, मुझे लगता है मेरे सारे जो 6 to 11th क्लास के स्टूडेंट हैं, जिन्होंने अभी लाइव देखा है या जो बाद में देखेंगे, उन सबके लिए एक बहुत ही प्रेरणादायी और रोचक जानकारीप्रद ये रहा है। और हम लोगों को ऐसी उम्मीद है कि आपसे हम लोगों को मार्गदर्शन, इस तरह के लेक्चर हमारे बच्चों के लिए और आने वाले समय में मिलता रहेगा। इसके साथ ही मैं उमेश क्षीरसागर जी, जो हमारे आईआईटी इंदौर के प्राध्यापक हैं, उनको मैं आग्रह करूंगा कि एक फॉर्मल वोट ऑफ थैंक्स के लिए। उमेश क्षीरसागर जी।
Good evening, sir. I would like to thank you a lot for agreeing and delivering talk here for our young students on molecules of life. I am very much confident that you have explained here very important secret of life, which is very interesting, simple, but same time it is very critical also and essential also. You have explained this in a very simplified manner so that every one student here not only understood this but also enjoyed this. We are really thank you for explaining this topic very simplified manner, sir. On the behalf of organizing committee, we again would like to thank you for agreeing and delivering this very important topic to our students. Thank you very much, sir.
Okay, Umesh ji, uh, thank you for this. I would like to invite Prashant ji to say few words, closing remark, so that we can proceed further.
Thank you, Brijesh ji. As you and Umesh ji rightly said, it was a wonderful opportunity for all of us to understand this into a very simple, non-technical language, and that too from a person like Dr. Mande. So I also join you guys to thank him again. Student internship is one of the unique platforms which is offered to BVM winners, national winners as well as zonal winners. This was a unique experience we started this year, and we will continue this further. All of you know that the upcoming national camp for this year's VVM exam will be there in ISAT Trivandrum, and winners of this national camp will be also participate in the Srijan internship. They're eligible for Bhaskara fellowship, and we are really excited on behalf of the core team, academic team, that some of you will be also representing your state in the national camp at Thiruvananthapuram. So with that, we will promise you that we will have many such exciting talks and physical internship which will be announced in the national camp itself. Thank you very much, and we'll be in touch with you. Thank you.
थैंक यू। अगले लेक्चर के लिए हम फिर से मिलेंगे। नमस्कार, धन्यवाद।